Our planet, a vibrant blue marble in the vastness of space, teems with life in every imaginable form. From invisible microbes to the mighty blue whale, Earth's diversity is astonishing, but how did it all begin? For centuries we've searched for the answer to this ultimate mystery. The Earth formed over 4 billion years ago, a hostile world bombarded by asteroids and wrapped in a thick alien atmosphere. Yet from this chaos the first simple life emerged. The leap from non-living chemistry to living biology is one of science's greatest puzzles. We know the early Earth had the right ingredients: water, carbon, nitrogen, and even complex organic molecules found on meteorites. The real challenge is understanding how these ingredients assembled into self-sustaining, evolving systems. By unraveling this mystery, we not only learn about our own origins but also gain clues for finding life elsewhere in the universe. At the heart of life's origin is a classic chicken and egg problem: which came first, proteins or the genetic instructions to make them. Proteins are essential for life, but the instructions for making them are stored in DNA and RNA. The machinery that reads these instructions is itself made of proteins and RNA, creating a loop of interdependence. Did proteins arise first, or did genetic molecules like RNA lead the way? Some theories suggest proteins or metabolic cycles came first, but the most popular is the RNA world hypothesis. RNA is unique; it can store genetic information and catalyze reactions, potentially solving the chicken and egg dilemma. In theory, an RNA-based life form could replicate itself and catalyze its own reactions. But a major question remains: how did life transition from this RNA world to the modern world, dominated by proteins and DNA? The missing link has long eluded scientists. A recent breakthrough published in Nature has reignited excitement in the scientific community about the origins of life. This new research offers a fresh perspective on the ancient puzzle of how life's building blocks first came together on Earth. Chemists led by Matthew Powner at University College London have demonstrated that simple protein fragments, known as peptides, could have formed with the assistance of RNA, long before the evolution of complex cellular machinery. Their findings suggest the existence of an intermediate RNA peptide world, a stage where RNA and amino acids began to interact and collaborate, setting the stage for more complex life. In this scenario, RNA acts as a molecular scaffold, gathering amino acids and linking them into short chains, far simpler than the intricate protein synthesis that happens in modern cells. This chemical partnership may have been the crucial bridge connecting the world of self-replicating RNA with the world of functional proteins. The researchers showed that, under conditions similar to those on early Earth, the formation of these peptides is not just possible, it may have been almost inevitable, given the right ingredients. No complex biology was needed; just the right molecules, a bit of water, and the right environment could spark this chemistry. This discovery fills a crucial gap in our understanding, revealing a plausible and testable path from simple chemistry to the emergence of the first living cells. For the first time, scientists have a tangible map connecting the mysterious RNA world to the protein world, bringing us closer to understanding how life truly began. To test their groundbreaking idea, the scientists set out to recreate the conditions of early Earth right inside their laboratory. They began with a simple water solution, carefully adjusted to a neutral pH, mimicking the ancient environment where life might have first emerged. Every detail was designed to reflect what our planet may have looked like billions of years ago, from the temperature to the chemical composition of the solution. Into this primordial mix, they introduced the key ingredients: short RNA fragments and a variety of amino acids, the essential building blocks of proteins. These molecules are thought to have played a crucial role in the earliest stages of life. But there was still a missing link: pantethine, a sulfur-containing molecule believed to have been present on the young planet. Its unique properties made it a likely candidate for facilitating early chemical reactions. When pantethine was mixed with amino acids, it triggered a remarkable reaction. The result was the formation of energy-rich compounds called amino-acyl thiols, which effectively activated the amino acids, making them ready for further chemical transformations. Next, these activated amino acids were combined with RNA. Amazingly, they spontaneously attached themselves to the RNA strand, a process known as aminoacylation. This is the very first step in the complex process of protein synthesis that all life relies on today. The RNA acted like a primitive assembly line, holding the amino acids close together and allowing them to link up into short chains called peptides. This simple mechanism hints at how the earliest proteins may have formed. What's truly astonishing is that all of this happened without the help of enzymes. No biological catalysts were needed. The process was driven purely by the inherent chemistry of the molecules themselves, showing how life could begin from simple reactions. The experiment proved that even in the absence of life, simple non-biological processes could create peptides on an RNA template, supporting the idea that life's building blocks can form naturally. It's a powerful demonstration of how the essential components of life could have assembled themselves, step by step, through natural processes alone. This evidence strongly supports the idea of an RNA peptide world, a crucial stepping stone in the journey from chemistry to biology, and from simple molecules to living systems. The experiment bridges the gap between random chemical reactions and the emergence of the first biological systems, offering a new perspective on how life might have started. It's a fascinating glimpse into how life's earliest partnerships, between molecules like RNA and amino acids, may have begun, setting the stage for everything that followed. The star of this new story is pantethine, a molecule few have heard of but possibly crucial for life's origin. Pantethine, related to vitamin B5 and a component of modern coenzyme A, likely existed on early Earth thanks to volcanic sulfur and simple organic chemistry. Its role is to activate amino acids, making them eager to bond with RNA. This chemical matchmaker enables amino acids and RNA to interact meaningfully, something they wouldn't do on their own. The process uses a high-energy bond like a compressed spring to drive the reaction. This strategy, using an activator to prepare building blocks, is common in biology today. The researchers believe that with amino acids, RNA, and pantethine in water, forming RNA peptide structures was almost inevitable. This suggests life's first steps weren't a fluke, but a likely outcome of early Earth chemistry. It paints a picture of a planet actively pushing toward life. Despite its promise, this chemistry has limits, especially where it could have happened. High concentrations of pantethine and other molecules are needed, making small enclosed ponds or lakes more likely cradles of life than the vast ocean. In a small pond, evaporation could concentrate the ingredients, increasing the chances of meaningful reactions. This supports Darwin's old idea of a warm little pond as life's birthplace. It contrasts with the deep-sea vent theory, which suggests life began in the ocean's mineral-rich fissures. The new findings tip the scales toward freshwater environments. If this chemistry was key, we should look for ancient lakebeds when searching for life's earliest traces. Life's origin may have been an intimate event in a quiet pond, not the open sea. There's a catch. The peptides formed in this process are random, not the precise sequences seen in modern proteins. Today, ribosomes build proteins with exact order, but early RNA scaffolds just grabbed whatever amino acids were nearby. The result: chaotic mixtures, not functional proteins. Most of these early peptides were probably useless, but nature works with huge numbers and vast timescales. Occasionally, a random peptide might have stabilized RNA or catalyzed a simple reaction, giving its RNA-peptide combo a survival edge. This is where natural selection could begin to act, favoring helpful combinations. Over time, certain RNA sequences might have attracted specific amino acids, reducing randomness. This marks the dawn of coevolution between RNA and proteins. The discovery doesn't solve the genetic code mystery, but it provides the raw material for evolution to shape. It's the first step from random chemistry to purposeful biology. The scientific community has greeted this research with excitement and healthy skepticism. Many praise its elegant, plausible solution to a long-standing problem and its demonstration under realistic prebiotic conditions. Critics note that the randomness of the peptides and the need for high concentrations of starting materials remain challenges. These are not dismissals, but invitations for further research. Scientists will now explore whether certain RNA sequences prefer specific amino acids and whether other activator molecules could work. This discovery isn't the final answer, but it's a vital clue, a foundation for a new model of life's origins. It brings us closer to understanding how non-life became life. This discovery opens a new chapter in our quest to understand life's beginnings. For centuries, humanity has gazed at the stars and wondered how life first emerged from the raw materials of our planet. Now, with each new finding, we peel back another layer of the mystery, revealing clues that have been hidden for billions of years. It moves the debate from theory to testable chemistry, offering a real mechanism for the partnership between nucleic acids and proteins. For the first time, we can recreate and observe the very reactions that may have sparked the dawn of life, transforming speculation into tangible science. If these steps are chemically inevitable, life may not be a rare accident, but a common outcome on planets with the right conditions. The universe could be teeming with life, waiting to be discovered, and our own story may be just one of countless others unfolding across the cosmos. Our search for life beyond Earth is guided by these insights, pointing us to freshwater environments and sulfur-based chemistry. These are the places where the building blocks of life might assemble, both on our planet and on distant worlds. This work reveals how complexity can emerge from simplicity and how life is a natural extension of the universe's laws. From the simplest molecules to the first living cells, the journey is one of increasing order and beauty, shaped by the forces of nature. The story of life's origin is our own story, connecting every living thing to a single ancient beginning. Every organism on Earth, from the tiniest microbe to the largest animal, shares this deep and remarkable heritage. By tracing these chemical pathways, we uncover the events that made our existence possible. Each experiment, each discovery, brings us closer to understanding the intricate dance of molecules that led to life. This discovery doesn't close the book but adds a hopeful new page to the greatest mystery of all. Where did we come from? The journey of exploration continues, fueled by curiosity and the promise of new revelations. And with each step, we move closer to the answer, guided by the light of discovery and the enduring human spirit to know our origins.